Hey everyone, hopefully you've already watched our first video where we went over beginnings of seed plants, talking about some of the different gymnosperms and touching on angiosperms. And now we'd like to go into the life cycle of gymnosperms. So this is going to be for activity 2 of your worksheet. So let's get started. Okay, so today we're going to use the pine tree as a model organism for exploring the life cycle and different parts of gymnosperms. Okay, so to start off, if you remember from the last video, I mentioned that our conifers are going to be monoecious. So that means they have both the female and male parts on their own plant. So the female cones are going to grow in the upper branches and the male cones in the lower branches of our diploid sporophyte. This is to prevent self-pollination. We want to, spread the genes elsewhere. The goal is not to pollinate on their own. So the female cones are located on the upper portions so that the male pollen from the male cones do not reach the upper portions of the tree where the female cones are. So here is a female cone, an immature one. So it's going to be green and very dense. They have a lot of moisture within them, and they will remain closed like this while the ovule develops. We don't want any pollen to get in there at this point. So here is going to be a more mature female cone where we see it begins to dry out and kind of open up so that pollen can come inside. And here, if you were to cut this open, you see our immature. So this is this side cut open. This is our immature ovules growing within. So here is looking at it a slightly different way. So each scale generally has an ovule with it. So here is a scale of the cone, and then here's our ovule within. You can see that even better here. Here's our scale, and then here's our ovule within. And each immature ovule will contain a megaspore. So here you see two fertile scales, here and here, and those contain our immature ovules, and each of those has a megaspore with it. So if you remember from seedless plants, we had the megasporangium and the microsporangium that created our megaspore and microspore. The ovule here is kind of like the megasporangium, creating the megaspore that will then produce our megagametophyte. So after meiosis of our megaspore mother cell, which is diploid, we are going to form our megaspore, which is haploid. So here is our megaspore. Through mitosis, the megaspore will then produce our megagametophyte tissue, which is what is labeled as number 2 here. That will then go to produce our archegonia, which is shown in part 3 here. So generally it forms 2 or 3 archegonia. Within the archegonia, we get the formation of 1 egg cell per archegonia. So here we only see it in one of the archegonia. I don't know why this one is missing it, but it is. So just to kind of bring this together a little bit more, so starting with part A, we see the megasporangium, which is this whole piece here, containing 1 megaspore mother cell, which is diploid. Through meiosis, we will get the production of 4 haploid megaspores, but only one of those is going to survive. So then through mitosis, that 1 surviving haploid megaspore will grow, and it will grow into the megagametophyte. The megagametophyte will then produce 2 to 3 archegonia here, which is going to house our eggs. Remember, there's 1 egg per archegonia here. And you may notice that we have this pollen grain that has been here for a while, all the way since we were still developing our haploid megaspores. So, the timing shouldn't have to be perfect. You shouldn't have to have a pollen grain know to land once we already have these archegonia created. We wouldn't get a lot of fertilization and reproduction that way. So the pollen grains can sit at the edge of the megasporangium for up to a year or even longer sometimes, waiting for the eggs to be developed and ready for fertilization. So here we see that pollen grain. Now that the eggs are ready, it extends out this pollen tube. And remember, the pollen grain contains our sperm. So then the sperm are able to go through the pollen tube into our archegonia and fertilize the egg. And then we see our nucleus form within our archegonia as well. So then once the egg is fertilized by the sperm, it becomes the embryo, which is then going to be diploid. Remember, when we go through fertilization, we're combining two haploid cells, and we're going to form our diploid embryo, which will grow in the nutritive tissues of our megagametophyte within the megasporangium, which is the whole piece here. And then once the embryo is mature, it will, with the female nutritive tissues or the megagametophyte, it will form a seed. So here we see once the ovules are mature, the cone will open up to allow that pollen grain to come in there to fertilize the egg. And at this point, the cone is going to be drier and a bit lighter as well. So once the egg is fertilized, it will become a zygote. And at this point, the ovule itself is going to be our seed. So here is a look at that seed that will come out of our cone. And remember the seed here, this is kind of like those little helicopter seeds that we played with as kids. We throw it up and it kind of comes down like a helicopter, perfect for flying it around. It's developed like that because our conifers, reproduce through wind a lot of the time. So they need to have a good structure to move through wind and land in other areas. So the main part of that helicopter, we have our actual seed that is at the base of that, and then the wing that allows it to move through the air. So one piece that's kind of cool is within the embryo that is developing, or around it, we have our tissues of the megagametophyte. So the megagametophyte tissues are going to serve as food for the seed. So, our embryo is still developing in here. It's not quite ready to come out and start forming the sporophyte. So in this time period that, it has landed and maybe is trying to grow further, now that it's in prime conditions for growth, it will continue to grow through this megagametophyte tissue as nutrition. So that was primarily the female side of things. Now we're going to look at the male side a little bit. So here are our male cones. You may be familiar with seeing these all over the ground late in the year. So our male cone, which is diploid, will have microsporangia, which are also diploid. The microsporangia will undergo meiosis to produce our microspores. Remember, anytime we're undergoing meiosis, we're going from diploid to haploid. So our microspores are haploid. The microspores will then undergo mitosis to form these air bladders that are going to help carry it through the air to find the female cone. And they also form the pollen tube through mitosis that are in that final pollen grain. And this is all haploid here. So now we're going to go through the whole life cycle with a pine tree. So we're starting with our mature sporophyte here that is diploid. On the male side of things, we have our male cones with the microsporangia that we just looked at. Through meiosis, we will form the microspores, which are then haploid, and that will form our pollen grain. On the other side of things, at the same time, we're forming our ovulate cones, which contain the ovules, and those contain megasporangia. The megasporangia contains our mother cell, which is diploid. And through meiosis, that mother cell will produce 4 meiotic products known as the megaspores, which are then going to be haploid. 3 of those are going to die, leaving only 1 megaspore to continue growing. So at this point, we have pollination occur. So we have our pollen grain that has flown through the air through wind and landed on the megasporangium. At this point, inside the megasporangium, we still have our megaspore developing, forming our archegonia, and therefore our eggs. This pollen that is attached will use the pollen tube to deliver the sperm to the egg, and through fertilization, we will form our embryo. The embryo will grow through mitosis, and remember this embryo is going to be diploid because we are combining our female haploid egg and our male haploid sperm to produce one diploid embryo. And this diploid embryo will continue growing within the megasporangia and turn into a seed. The seed will fly through the air, and when it lands in prime conditions, will begin to grow into the developing sporophyte and eventually become our mature sporophyte that is diploid still, and then the process starts over again. So we just went through the gymnosperm life cycle using the pine tree as an example. So you can now do activity 2 for your worksheet, and I think that's everything. So we will start back with the next video soon.